A new wave of optimism is beginning to take shape in the long and often disheartening battle against Alzheimer's disease. Researchers at the University of New Mexico Health Sciences Center have made a significant leap forward with the development of a tau-targeting vaccine that has successfully passed the critical preclinical hurdle. With robust results from studies involving both mice and non-human primates, scientists are now preparing to move toward human trials, a step that, if successful, could redefine the future of Alzheimer's prevention and treatment. Unlike traditional approaches that have focused on amyloid beta, the hallmark plaque-forming protein most commonly associated with Alzheimer's, this vaccine targets the tau protein, which is increasingly recognized as a more decisive factor in disease progression. In Alzheimer's, tau undergoes pathological changes, most notably a modification called phosphorylation, that transform its role from stabilizer of neurons to a toxic agent. When tau detaches from microtubules and forms tangles in the brain, neurons are unable to communicate effectively, leading to the cognitive decline that defines the disease. By targeting the phosphorylated tau region, known as point 181, researchers are not just treating symptoms, they are potentially stopping the process that drives neuronal dysfunction at its core. In previous studies, including one published in 2019, the vaccine generated clear and measurable cognitive benefits in mice genetically engineered to express pathological tau. The results were not just biochemical. They translated into better memory and cognitive function in behavioral tests. Now, with this latest research, scientists have expanded their scope to include 2 additional strains of mice, one of which carries the human tau gene, as well as macaques, non-human primates whose physiology more closely mirrors that of humans. In each case, the vaccine triggered a powerful immune response, and the antibodies produced were able to bind effectively to the same form of tau found in human patients. Equally encouraging is the technology used to create this vaccine. It employs virus-like particles, VLPs, a platform that mimics the structural properties of viruses without their infectious DNA. These particles are decorated with small fragments of the tau protein, specifically the phosphorylated point 181 segment, which the immune system can easily recognize and attack. This method allows for the generation of long-lasting immunity with just one initial injection followed by 2 boosters. It also bypasses the need for adjuvants, chemicals that are often added to vaccines to enhance immune response but can sometimes cause side effects. VLP-based vaccines have already shown a strong safety profile in humans, increasing confidence that this Alzheimer's vaccine may also prove to be both safe and effective. The research team did not stop at preclinical immune response testing. They also examined whether the antibodies generated in immunized animals would recognize human tau protein. Using blood samples and brain tissue from individuals with mild cognitive impairment and Alzheimer's disease, they confirmed that the antibodies produced in vaccinated monkeys could indeed bind to the human form of pathological tau. This cross-species reactivity adds an important layer of validation, suggesting that the immune mechanisms observed in primates may also occur in humans. Another strength of this development lies in its collaborative foundation. The work brings together expertise from UNM, the University of California, Davis, and the California National Primate Research Center, institutions that lend considerable credibility to the findings and provide the infrastructure necessary for rigorous scientific validation. This kind of multi-institutional support is essential for transitioning a vaccine from animal studies into clinical human trials. Though funding is still being sought for the initial phase 1 trial, there is every reason to believe that support will follow. The Alzheimer's field has long been in search of innovative therapeutic approaches, especially those that offer more than the modest benefits seen in amyloid-targeting drugs. By moving further upstream in the disease process, targeting tau directly, this vaccine could represent a paradigm shift, not only in treatment, but also in prevention. What sets this work apart is its forward-looking potential. If the vaccine proves effective in humans, it could be administered years before symptoms appear, particularly in individuals with known genetic risk factors or early signs of tau accumulation detected through biomarkers. This would mark a transformative moment in neurology, enabling clinicians to act proactively rather than reactively, reducing the devastating burden that Alzheimer's imposes on patients, families, and healthcare systems. In the context of a disease that currently has no cure and only limited treatment options, This tau-targeting vaccine represents more than just scientific progress. It represents hope. Hope that the future of Alzheimer's care may no longer be confined to symptom management and late-stage interventions. Hope that we may be standing on the threshold of a new era where Alzheimer's can be prevented before it takes hold, sparing millions from its slow, heartbreaking course. While many hurdles remain, the foundation laid by this research is solid, and the horizon ahead has never looked more promising.